I'm thrilled to introduce my friend Chris Kahle. Chris is an assistant professor of neurosurgery, pediatrics, and cellular molecular physiology at Yale, where he completed his MD PhD before he did a residency and fellowship in pediatric neurosurgery at Harvard. Chris is one of the most active and successful researchers currently in pediatric neurosurgery. He works on one hand on the genetics of congenital malformations and has published in very high impact journals on, for example, the genetics of hydrocephalus in Nature Medicine, the genetics of Valent-Galen, malformations in neuron, and autism and science signaling. He also runs a basic science lab in which he studies ionic and water homeostasis in the developing nervous system, including hydrocephalus. Among others, this work has the potential of identifying drug targets to prevent or treat these disorders. Chris is from Milwaukee and had planned to visit us on one of his trips to see his parents, but COVID somehow got in the way, as it has in so many other ways. But we're delighted to have him give us at least a virtual lecture. He's a nice person with a lot to teach. Thank you. Thanks, Chris.
Thanks a lot, Benny. Thanks a lot, Dr. Dempsey. I'll get to the specifics here in a minute. Benny, for sharing the screen, we usually do most of our stuff on Zoom. I'm sure it's easy here. What's the best way to share my presentation?
So Chad is on. He'll give you specific directions.
Okay. Chad, can you help out?
Yeah, so down at the bottom of the screen, you should have a toolbar down there.
Yep.
There's the third icon, I believe it is a kind of up arrow.
Yeah, open system preferences.
Nope, it should be share content.
Share content. Maybe see if more options here.
The toolbar only appears if you hover just at the bottom of your screen. And you'll see like mute and turn your camera on, things like that.
Yeah, mute, stop my video. Third one, system preferences. Fourth, close participants chat, more options.
He doesn't have shared content in the chat. Can
Okay.
you get that to him, make him a host or something of that nature?
Change role to presenter. Okay, now you might have the capability.
Okay, it says I'm the presenter now. And let's see here. Stop my video. Okay, it says if I go to open system preferences, it says allow WebEx meetings to record your computer screen so that you can share content in meetings. Would that
Yep, you'll need to select that.
Okay, let's try that. Open system preferences. And let's see, what would I pick here? PowerPoint, presumably?
No, there should be a WebEx checkbox that you guys select. I assume you're on a Mac?
Yeah, I'm on a Mac.
Yeah, so are you in the system preferences, security and privacy?
Yep.
Tab, and then you gotta scroll down to screen recording, I believe.
Yep, screen recording.
And then check on Cisco WebEx meetings.
All right, clicking on that now. Cisco web meetings will not be able to record the contents of your screen until it is quit. Quit now?
Okay. Yeah, you might have to disconnect and reconnect quick. Sorry.
All right, sorry. Leave meeting. All right, you guys can hear me again?
Yep.
All right.
I'm making you the presenter again.
Okay, then I've got now a new icon that says share content.
Yep, that's what you need.
All right, hitting that now, and then Microsoft PowerPoint,
Yep.
share. And you guys can see that.
Yep.
All right, I'm going to view it as a slideshow now to make it there.
Perfect.
And are you able to see it?
Yes.
All right, good. All right, so I'm sorry for that annoying delay, guys. use a different program here. I won't take up too much of your time. I realize, as well as anybody, the necessity of getting to the OR on time. So we'll dig right into it. I was invited to talk a little bit about our work, mainly in the lab, but the overwhelming message that I'm going to try to get across now is how, number one, neurosurgeons and neurosurgeon scientists can make a real contribution, and number two, how that contribution can be very translational and very meaningful for patient care. As a MD PhD student who was trained at Yale and then went up to Boston to do clinical training, especially during residency, I found it a bit disorienting, and it's easy to lose vision if you're somebody who was trained in a science background and then are immersed in clinical training. The question being, is it possible to do real science as a neurosurgeon, and what kind of what are the contributions that one can make in that regard? And I would really emphasize that there is a huge role for a neurosurgeon who brings specific clinical insights to scientific questions. And if you're in the right place at the right time with generous, knowledgeable colleagues like Benny, for example, who's really helped me out on the hydrocephalus front, Beverly with the vena Galen malformation front, you can make some real serious inroads. So today we'll focus on the hydrocephalus story, but we've got multiple projects in the lab that all fall under the same general problem, which we're trying to answer, which is, what are the genetic contributions to many of the pediatric neurosurgical disorders that we treat? And can knowledge of those genetic determinants help us reclassify disease and change the way in we think about how to treat patients? So bringing up, this scan, this is from one of my own patients. Commonly, new patient in the emergency room comes to your care. As a resident, you're called down. This is the scan. And the question is, what's your plan? And when you look at this, I think our field has been traditionally conditioned to think of hydrocephalus as a fluid problem. And certainly in a lot of cases, certainly in acquired hydrocephalus, this is absolutely the case, where there's an impairment in so-called brain plumbing with an accumulation of CSF, and the way that you treat it, given that paradigm, is with a shunt. But what I'd like to get across here, and I think that this is something that anybody who's spent time with these sort of patients knows, is that not all hydrocephalus is created equal, and that congenital versus the various forms of acquired, there can be some radical differences. And so our questions, the question that I was interested in answering is what are those differences, and what implications might that have for treatment? These are some of my disclosures. We're a well-funded NIH lab with 3 primary R01s at this point in time, mainly centered on the genetics of pediatric neurosurgical diseases and the mechanisms, and then some foundation funding. Acknowledgements up front, just so that if we run out of time, the people who are doing the work and the students get the credit, a lot of them coming down the pipeline for neurosurgery residency. I think our field has a very unique position, in that a lot of the people going into it, for good or for bad, have a extremely driven, ambitious, but highly competent nature. And so one thing that I've been incredibly blessed with at Yale is by folks even taking a 5th year in medical school, taking some time off in lab, who make real meaningful contributions. So some of this work is not only done by PhD graduate students, but people in the MSTP program, but also 5th year medical students, people making big contributions by bringing energy in a focused way for that 5th year. And wanting to acknowledge, as I have remarked already, about our collaborating physicians and institutions like UW, like Benny, like Bev. Dr. Dempsey has really been a kind of an example for me of what I would like to get to someday. I've gotten the Dempsey Award 2 times as a resident, and has these guys said I'm from Milwaukee, went to Marquette High, Jesuit school down in Milwaukee, and my family is still in Wisconsin. So this is kind of a special day for me. Quickly introduce the concept of kairos for all the Greek philosophers or theologians out there. It's a Greek word for time, but not time as we usually think about it. Chronos, the other Greek word for time, talks about the time on a clock, but kairos really talks about a timing or an opportune moment. Time for action. And so when I was thinking, making the transition from residency and fellowship down to faculty, one thing that my mentors taught me is to think about at the specific, place and time that you find yourself, what might you have privileged insight into, or resources that stack the deck in your favor to be able to answer a specific question. And so for me, landing at Yale for a couple of different reasons, including my previous history there and my recruitment and what I thought were some strengths of the institution, both from a neurosurgery and basic standpoint, that would be very different from if I stayed up in Boston at Boston Children's, or went to Wisconsin, someplace else. You can only do with what you have where you are. And so for me, surgically, is hydrocephalus my favorite thing in the world? No, it's not. I love, juicy PFOSA tumor. Although I love tackling hydrocephalus with endoscopic approaches and so forth, but I like a lot of the complex tumor surgeries but when from a scientific standpoint, I was a bit intimidated about getting into cancer biology. I thought it was saturated, and when I looked forward to the future, and especially given some of the diseases that are under our belt as pediatric neurosurgeons, I thought my training in human and molecular genetics, coupled with our lack of knowledge about a ton of the diseases that we treat, and then also where the field was with in terms of technology with the advent of next generation sequencing, whole exome sequencing, I thought that it was the right and opportune time to venture into the neurodevelopmental side of our diseases with a lot of low-hanging fruit. And so also the other concept of when you're thinking about what to do or what to study, one of my pastors growing up in Wisconsin sent this quote along to me, and I've never forgotten it, by Frederick Buechner. He's a Methodist preacher who said, "Vocation is the place where your deep gladness meets the world's great need." So to hear that calling, that vocation, which I believe we all are called to do, you first of all need to define what you're passionate about, what you like doing, and then open your eyes to the needs of those that you encounter and try to find a match. So the question is as a neurosurgeon, what can you uniquely bring to the table to help fill that knowledge gap or heal that person or fill that need? So historically, our field in neurosurgery and in pediatric neurosurgery, if you think about it, does some pretty crazy things to the brain in a sometimes maximally invasive fashion. If you look on the left, you see a swollen cortex malignant cerebral edema in the setting of stroke, and to address that, we do a decompressive hemiocraniectomy. In the middle panel, of course, you see a hydrocephalus associated with a choroid plexus papilloma, and to treat hydro, often we stick a ventricular catheter into the fluid-filled space. Not so much now, but certainly in the past, we've addressed medically intractable epilepsy by taking massive pieces of brain out. The point of this slide is really just to say that each one of these processes is driven by molecular pathophysiology events that lead to a certain manifestation of disease that we treat in the best way we know how. But I think it's obvious to say that if we had a better understanding of the molecular pathophysiology, there might be less invasive, more efficacious ways of treating these things. So this is a slide my mentor, Rick Lifton, gave me when I was doing my PhD in genetics at Yale. And this was really to, in an absurd way, emphasize how complex homeostatic systems are. So this wiring diagram from a famous figure that the renowned physiologist Guyton made in 1972 about all the regulatory components of blood pressure regulation could be applied to any complex physiologic system in which there are inputs, outputs, regulatory nodes. And it's just to show that if you're interested in studying a problem, so for example, cerebral spinal fluid homeostasis and how that might lead to a disease like hydrocephalus, the question is, in all the different components within that system that are responsible for maintaining homeostasis, which one is the most powerful, or which one could be targetable to influence behavior of the total system for therapeutic effect? You know, many times we go into these problems with a hypothesis, and of course that can be good, but it can, that hypothesis in and of itself can corner you in a way that prevents you from discovering those truly targetable nodes down to the amino acid residue level that by manipulating them might have a more profound effect on system behavior. And so genetics classically has been a terrific unbiased way to open your eyes to the different possibilities within that complex labyrinth that I just showed you, to identify targetable points that are not only important for behavior of that physiologic system, but that might be targeted for therapeutic effect. And so traditionally, rare familial forms, large family pedigrees using positional cloning were used to identify single gene mutations. But more recently, and this is really something we benefited from because of next generation sequencing, is that we can take even sporadic cases in what we call case parent trios, mom, biological dad, and the affected proband, and find de novo mutations that aren't inherited from mom or dad, but arise during development. This approach is a well-known approach to identifying critical targets. And even if these are rare, damaging mutations, they have large effect and may be more relevant for more common diseases. So for example, everyone knows Lipitor and Repatha. A bunch of us are probably on them right now. But these drugs target genes and gene products that were discovered through the investigation of rare Mendelian forms of disease, in this case, hypercholesterolemia. So the idea that by studying rare forms of disease, you can understand pathophysiology and identify targets that might be more relevant for more common forms. We've used this approach with some success, as Benny has said, and not pictured here is the new publication that will be coming out in about 4 to 6 weeks in Nature Medicine, where we take our second take on hydrocephalus genomics, finding 8 new genes. So hydrocephalus, really don't have to belabor this point at all, but to say that the kind of classic paradigms of hydrocephalus emphasize an accumulation of cerebrospinal fluid in the brain ventricles that leads to increased pressure, can lead to coma and death because of that. If it's untreated, it's common. If you're a pediatric neurosurgeon, it's the bread and butter of your existence, for good or for bad. And so the fact is that our current classification system of hydrocephalus is antiquated and incomplete. I think any neurosurgeon and scientist would agree with that. Generally speaking, hydro, primary and secondary. Secondary, we're not going to talk about today, although we have projects going on in that that are fascinating and more related to ion transport and choroid plexus function. Those sorts of forms of hydrocephalus are usually in the context of infection or hemorrhage, sometimes tumor. They can be obstructive, not obstructive. That has ramifications for how we treat hydrocephalus, whether it's endoscopy or a shunt. But in congenital hydrocephalus or developmental hydrocephalus, basically that form of hydrocephalus for which we have no other explanation and which can be seen even at birth or shortly thereafter, we still think of it in terms of these communicating versus obstructive pathophysiologies from a very pragmatic way. And that has, real implications for whether we choose to do an endoscopic third ventriculostomy or, use a ProGav adjustable shunt. The treatment, we usually, when you're writing a grant for hydrocephalus, you kind of bemoan the existence of shunts and talk about our need for better treatments, and that is certainly true. But the fact is that of the procedures that we do that make a real difference, shunts are one of the biggest innovations that have happened in neurosurgery in the last 50 years. Despite that, it is the #1 cause of medical device failure in the United States, and it leads to an incredible economic, social, physical burden for both patients and their families. So new treatments, are absolutely required. This is especially the case if you're working with children in places in the world where there aren't a lot of neurosurgeons or the capacity and funds for close follow-up and placing hardware in the operating room. I had the luxury and privilege of training with Ben Morf during my time at Boston Children's and learned endoscopy from him. And so the concept of trying to find innovative ways to treat hydrocephalus beyond current shunting paradigms is one I think that catalyzed my interest in discovering or trying to discover the come to a deeper understanding of what the drivers of congenital hydrocephalus are. So addressing these gaps and these needs would have an obvious benefit. It would decrease patient morbidity and mortality. It would improve the neurodevelopmental health of the population and benefit millions of children and their families worldwide, especially in those places where these resources are scarce. To understand the pathophysiology of congenital hydrocephalus, people have traditionally looked at these large pedigrees, which are extremely useful, and they've led to the discovery of L1-CAM and MPDZ, but these account for a very small fraction of the patients that we treat. And despite that, there's been very good epidemiologic studies that show that still a significant proportion of our congenital hydrocephalus cases are predicted to have a genetic etiology. So it just really suggests that many more congenital hydrocephalus genes await discovery, and coupled with the technology that's now available, that really drove us to get excited and involved in this problem. in addition to those barriers which I just mentioned, there are other nuances from a genetic standpoint that make gene discovery in this context difficult. They include locus heterogeneity, phenotypic complexity. You know, a lot of these kids have other stuff going on, whether it's other structural brain defects, whether it's intellectual disability, seizures, other developmental anomalies. So it's a complex phenotype, and so the term hydrocephalus really doesn't do justice to these sorts of realities that we experience as neurosurgeons with boots on the ground. And so, our hypothesis really was that this term, congenital hydrocephalus, after application of next generation sequencing technologies and deep clinical phenotyping, which includes neuroradiographic phenotyping, and which I would suggest that only a neurosurgeon could bring to the table, that this blanket kind of garbage term hydrocephalus kind of begins to break apart into specific sub-bins, where then we begin to understand that this type of patient with hydrocephalus has this sort of prognosis and should be treated this way versus this other patient with hydrocephalus, even if their MRIs look very similar. Having that genetic knowledge might be able to, it might enable us to come up with a new classification scheme. And so by using whole exome sequencing in this context, it's very powerful because it's unbiased, it's systematic, and it's, in some sense, this isn't a really novel approach. People have, my predecessors, including those that I trained with in my lab, like Murat Ganel and his approach to some of the aneurysm genetics, Matt State, who's also from the Lifton lab, who has applied this approach to autism. I'm just, I consider our lab and other labs like our lab as just kind of following the great examples of these guys and women who've gone before us. So I've talked about the importance of gene discovery. The one that I'll emphasize here, where we talk about it, maybe certainly write it down when we're trying to make our case for a funding for our project. But whether or not we really believe it, could there be a precision medicine therapy for hydrocephalus? And I would say yes, absolutely. And my point is that other fields who have made investment for discovering the genetic etiologies of the disease that they are responsible for caring for in their patients are now reaping the benefits. So these are disorders with single gene disorders where gene replacement therapy or other forms of gene therapy are bearing fruit in real time. So this is not something that is impossible or a pie in the sky for congenital hydrocephalus, depending on the mutation type and the developmental time points at which the disease manifests. So we set up this thing called the HydroSeq platform, of which University of Wisconsin and Benny is a part of, and our objective was and still is to use a functional genomics approach to discover new congenital hydrocephalus genes and to gain mechanistic insights into the discovered mutations. Three basic aims, and these aims for HydroSeq are really transferable to other diseases. So, for example, we have other things going on for Faena-Galen malformation and for other, AVMs, for other structural brain disorders like arachnoid cyst, which we're finding some extremely fascinating things for, and even a few diseases that, or conditions rather, that that are not in pediatric neurosurgery, but I find personally fascinating, and we have a paper coming out in a couple of weeks that shows that there's a small subset of trigeminal neuralgia patients, MVD resistant, that are, a component of their disease and probably the reason why they're medically intractable and surgically intractable are because they have rare large effect mutations in GABA signaling genes and a couple of calcium transporters that likely operate in the trigeminal ganglion. And so in that setting, the idea is, well, maybe if we know information like that, we might keep somebody from going and getting bilateral MVDs or repeat MVDs on the same side, or maybe it might help influence the choice of a different medication. But for all these different, diseases, you need to have a large, well-characterized, deeply phenotyped cohort. I think that is one of the big things that neurosurgeons, along with our collaborating neuroradiologists, can bring to the table. Then the genetics comes into play, and you have to. For us, we're blessed by the Yale Centers for Genome Analysis, the Centers for Mendelian Genomics at Yale. I think one of the premier genomic institutions in the country. UW is very strong in this. Seattle, the folks at UW, the other UW out in Seattle are very strong at this. Down in Texas, at Baylor, but we've been really blessed at Yale to have a robust sequencing platform. And then importantly, there should be, I would propose, a at least medium throughput way to functionally validate and gain some initial mechanistic insight into your discovered genes. So we have a nice Xenopus platform that is cheaper and more efficient than in mice, which we also use to model our discovered disease genes. But that gets really expensive and a bit laborious. So our approach is interdisciplinary. It brings a bunch of different people together, all that look at a problem from multiple different ways. It's an international dynamic recruitment platform with that has both kind of the classic investigator, institutional driven approach, but also something that has been highly advantageous to us is a family-driven social media campaign that took a while to get set up with our legal folks at Yale and the HIC committee, but we've recruited a bunch of patients through social media, and that has been extremely efficient and has led, I think, probably one of the biggest reasons why we've been able to move quickly on this problem. And then, of course, as I alluded to in one of the first slides, the timing is right for looking at the genetic determinants of some of these diseases that historically have been not looked at, primarily because they're under the care of neurosurgeons who might not, be well versed in genetics or maybe have an interest or knowledge that these approaches exist. But with whole exome sequencing, whole genome sequencing, and then some other functional technologies that we use, the timing is really, I think, right for this problem. And so through social media, these are some of the students who are that help out in the lab, including Adam, Hannah, Will, Stephanie, and Sierra. We've collected, which is probably the largest cohort to date with congenital hydrocephalus, and the cases really come in literally each day into the lab. This is the breadth of our recruitment outreach. You can see some of the big circles in Wisconsin there. But the social media approach through Facebook and Twitter has really helped us along. For sake of time, I'm not going to go through the protocol of our whole exome sequencing, but by this time it's pretty formulaic and certainly robust and repeatable. And for the last couple minutes, I'm just going to hit on some of the major points about some of the specific genes that we found, and perhaps more importantly, what this might mean. The next paper coming out, like I said, in about 4 to 6 weeks in Nature Medicine. Will kind of detail this, and the neuron paper before it kind of sets the table. But the fact is that we found that de novo mutations in about 6 to 8 genes account for roughly 20% of congenital hydrocephalus. On the one hand, that to me is pretty impressive, and certainly I think the most fascinating thing about these genes is that none of them are really or none of them are in genes that I would have initially hypothesized as playing a major role in congenital hydrocephalus. I came into the problem thinking that, given my background in ion transport from some genes that we discovered as a PhD student, that congenital hydrocephalus would be one in which we'd find these activating mutations and ion channels or transporters in the choroid plexus epithelium, and that through relative increase in CSF secretion relative to reabsorption or something like that, you could account for a bunch of cases. This isn't the case at all, but mutations in molecules like TRIM71 and SMARC-C1, these are very basic genes expressed incredibly early in development and important for maintaining neural stem cell pluripotency, for driving transcriptional programs that are important for neural stem cell differentiation, and then ultimately functional maturation of the of structures like the multiciliated ependyma. So this is just to show that mutations, it's obviously uninterpretable, but just to show you that mutations in genes that are highly expressed early in brain development are significantly enriched in congenital hydrocephalus patients relative to controls. This is a fascinating family in TRIM71 that mutated in this gene called TRIM71, which is regulates the RNAs by binding to them and degrading them. It's these are on the left two different patients, two different parts of the world that were shunted and were thought to require shunting. You can see just how closely these are unrelated patients. Look at how similar their MRIs are. Not only the ventriculomegaly, but also other structural brain defects. So the question is some of these cases that are deemed so-called congenital hydrocephalus and shunted, these are two different neurosurgeons who made these surgical decisions, and apparently the patients require CSF diversion. So the real question is how do you make, going back to my first slide, how do you make the decision whether or not to shunt, and how do you know what the prognosis of these patients are? But this example shows that this de novo mutation occurred, was passed on to a subsequent generation, and both these patients, which are not illustrated here, these are just different cases, had shunt-dependent hydrocephalus. This just shows you that some of the mutations that we discovered in TRIM71 are recurrent mutations, meaning that they occur at the exact same residue. These two residues are the ones involved. They occur in these things called NHL domains, which bind target RNAs and lead to their degradation. This is to show that these patients with TRIM71 mutations have similar clinical and neuroradiographic phenotype, suggesting it's a syndrome or a cluster of phenotypes associated with a specific gene mutation. TRIM71 is highly expressed in the embryonic brain, in the in neural stem cells that line the ventricular zone, and dramatically drops off with subsequent development. We've gone on to show that not only can you recapitulate hydrocephalus by engineering the specific human patient mutation into a mouse, but that the reason that TRIM71 leads to the massive ventriculomegaly that you see is not due to aqueductal obstruction, but it's really a impact on neural stem cell proliferation and ultimate differentiation. So a major paradigm I would like to emphasize is that I think as neurosurgeons, we're trained to hone in on the T2 bright signal of CSF in these hydrocephalic conditions. And what we're finding is that while that ventriculomegaly is certainly present, it may be more of a reflection of an intrinsic genetically encoded impairment of brain parenchymal development and less a primary disease of CSF homeostasis. The CSF is really accommodating itself to a vacuous container that is rendered as such by a primary impairment of brain development, defective neurogenesis, cortical thinning. We see that in subgroups of patients where after you place a shunt, the cortex does not expand. There's, in fact, not real high pressure. That doesn't necessarily mean that folks aren't necessarily dependent or benefit from CSF diversion, but I think a general theme is that what we're recognizing, and our study is highly attuned to detect things like this because we take a shotgun approach to patient recruitment. We're just not looking at patients that have been properly diagnosed and treated at UW or, Boston Children's or Yale. What we're doing is seeing, okay, how is hydrocephalus, congenital hydrocephalus, really treated worldwide and in the community? And what we're finding is that, folks with big ventricles, right, and with neurodevelopmental delay, other things that lower the threshold for intervening in these kids, we're probably over shunting some children with what we deem as congenital hydrocephalus or developmental ventriculomegaly. This is another version of hydrocephalus that we continue to find, and that's related to mutations in SMARC-C1, a chromatin modifying a protein in the SWE/SNF complex first discovered in yeast. So again, I'm picking out some extreme examples of patients with these mutations. Other patients with the SMARC-C1 mutations just have classical ventriculomegaly. But in this case, in contrast to the TRIM71 mutations, patients with TRIM71 mutations, all these folks have aqueductal stenosis. So these guys are really reliant on shunting. Their ventricular morphology and brain dysmorphology sometimes precludes them from being treated with endoscopy. But just like TRIM71 patients, we're finding recurrent mutations in SMARC-C1, similar constellations of clinical phenotypes. And people have done the beauty of genetics in this regard is that we've been able to look back in the literature, and people demonstrated even before we found mutations in humans that mutations in this gene have already caused hydrocephalus in mice. So it's a great proof of principle in a model organism, and you don't even have to do another experiment. Another pathway that comes up, and this will be the last remark I'll make about specific genes, is those in PIK3CA, mTOR, and PTEN. These have been previously implicated in brain growth or brain overgrowth disorders, other heterotopias that rarely can be associated with hydrocephalus, but we're finding that de novo mutations without those other overgrowth phenomenon are coming up time and time again. And the reason that we're kind of interested in the PTEN de novo mutations, which lead to PTEN loss of function, upregulation of mTOR and PI3 kinase signaling, is that Tyrone, one of our MD PhD students, will be, I'm sure, applying to neurosurgery in a couple of years. We've shown that treating mice with rapamycin, mTOR inhibitors, can reverse the ventriculomegaly and also address some of the seizures that's associated with this hydrocephalic phenotype. So that's kind of underlying the concept of, if we know what's causing the congenital hydrocephalus, we might be able to target it. I know my overrunning my time here, but this is to say that we found, and this will be in the upcoming paper, that there's some significant genetic overlap between what people have found with autism spectrum and what we're now finding with a congenital hydrocephalus, and that might help explain some of the associated neurodevelopmental phenotypes that as neurosurgeons, and I'm just speaking personally, we tend to not worry about too much, and we think that, okay, this, our threshold, if you think about it, for treating with these kids with shunts is actually quite low, in that, as long as the shunt's working, the kid is surviving, we kind of think about these other phenotypes as less important, but they're all potentially related to the same genetic driver and event. I'm not going to get into our functional platform that we use to validate these disease genes, but it's pretty cool. It's in Xenopus embryos, and that gives us an efficient way of seeing how these things can actually impair CSF dynamics or brain development at time points that you wouldn't be able to look at in mice. This just goes to show that, fascinating, at least to me, who spends time with this, that you can recapitulate aqueductal stenosis in a frog, and which leads to ventriculomegaly in a frog by engineering in via CRISPR a mutation that we found in a human patient. So definitely sounds weird, but thinking of creating frog avatars of specific human patients, and thinking of things like, okay, let's do a drug screen for this particular patient, or can we gain insight into this particular patient's pathophysiology by studying this lower organism with the same mutation. This is the platform that we've set up, that we're trying to mimic in certain other diseases, and I feel like we've only hit the tip of the iceberg with hydrocephalus. So in conclusion, some of our recent work and that of others has shown that de novo mutations in a relatively limited number of genes can cause up to 20% of sporadic congenital hydro, and that at least surprising to us, that all these genes are we found and others have found to be critical regulators of neural stem cell growth and fate, and not necessarily components of the glymphatic system or cerebrospinal fluid conducting transporters in the choroid plexus, or multiciliated ependymal components. These are more fundamental brain development genes. And so this really implicates impaired neurogenesis over a primary CSF overaccumulation, at least in a subset of patients. This is maybe a quarter of patients. And how could these insights impact clinical care? We're thinking about how, with the continued sequencing of more patients, collecting all the different MRIs of these folks, combining genotype phenotype correlations, even with machine learning, of how to predict what someone's genetic defect is from looking at their MRI. You saw how some of these MRIs are extremely similar. We think that we can come up with a more coherent and useful classification system that might allow us as pediatric neurosurgeons, when someone comes in through the emergency room, to say, hey, you know what, this patient has big ventriculomegaly, but because of this genetic defect, maybe shunting isn't the right answer here, or maybe it is for the patient to be, to have continued survival, but these are the other associated phenotypes that we can expect not to benefit from shunting, so that when the patient comes to the emergency room the next time, for symptoms that may or may not be related to the shunt, our threshold is raised for taking them back to the operating room to do a revision. So I would propose that this kind of stuff has very practical, real world application that I would hope we get to with, as our group and other groups continue to make efforts at Gene Discovery. So I wanted to thank you for your time and this invitation, and most of all your, Benny's contribution to the two studies that we've done.
Thank you very much, Chris. I really want to thank you for this mindset that you follow the science to change your paradigm or your thinking about the disease. And the real take home I got was this idea of looking at brain development at the primary stem cell level, which makes perfect sense. But it also implies probably 2 or 3 different types of potential therapy in the future. Well done. Thank you.
Thank you very much.
Other questions?
Chris, this is terrific, as always. A practical question. I mean, you said that you've been primarily following a shotgun approach so far, but have you tried to look at, for example, congenital aqueductal stenosis patients who've succeeded or failed ETV and compare their genetic makeup?
Yeah, I mean, these are the sorts of correlations that we envision as being fruit of continued sequencing and deep phenotyping. The fact is that, we might have the largest cohort, but to make these sorts of correlations, especially with a rare disease, you just need a lot of patience. Mm-hmm. And so that gives us, motivation to not only collect more patients, but then to go back and solve patients and families that we have, which we haven't on the first run been able to define. We've got maybe 22 multi-generational families that we still that clearly have a genetic etiology for their disease that using whole exome sequencing we have not been able to discover. And so that gives us motivation for applying other DNA sequencing technologies like whole genome sequencing to look in recognition of the fact that with whole exome sequencing we're biasing ourselves to a minute fraction of the entire genome, the exonic regions. So I have no doubt that as we collect more patients, use these other DNA sequencing technologies that will define these other sorts of hydro, including the ones that you just spoke about, and we'll be able to make those correlations. To me, that's the goal of the entire project. Thanks for the opportunity, guys.